Here we will discuss about necroptosis. Necroptosis is a programmed form of necrosis or inflammatory cell death. Conventionally, necrosis is associated with unprogrammed cell death resulting from cellular damage or infiltration by pathogens, in contrast to orderly programmed cell death via apoptosis. The discovery of necroptosis showed that cells can execute necrosis in a programmed fashion and that apoptosis is not always the preferred form of cell death. Furthermore, the immunogenic nature of necroptotic factors, its participation in certain circumstances, such as aiding in defense against pathogens by the immune system. Necroptosis is well defined as viral defense mechanism allowing the cell to undergo cellular suicide in a caspase independent fashion in the presence of viral caspase inhibitors to restrict viral replication. In addition to being a response to disease, necroptosis has also been characterized as a component of inflammatory diseases such as Crohn's disease, pancreatitis, and myocardial infarction. The signaling pathway responsible for carrying out necroptosis is generally understood. TNF alpha leads to stimulation of its TNF R1 receptor. TNF R1 binding protein, TNFR associated death domain protein, that is TRAD, and TNF receptor associated factor 2 TRAF signals to RIPK1, which recruits RIPK3, forming the necrosome, also named ripoptosome. Phosphorylation of MLKL by the ripoptosome drives oligomerization of MLK1, allowing MLKL to insert into and permeabilize plasma membrane and organelles. Integration of MLKL leads to the inflammatory phenotype and release of damage associated molecular patterns which elicit immune responses. So, we'll see the medical relevance of necroptosis. Necroptosis has been implicated in the pathology of many types of acute tissue damage, including myocardial infarction, stroke, ischemia, reperfusion injury. In addition, necroptosis is noted to contribute to atherosclerosis, pancreatitis, inflammatory bowel disease, neurodegeneration, and some cancers. In solid organ transplantation, ischemia reperfusion injury can occur when blood returns to tissue for the first time in the transplant recipient. A major contributor to tissue damage results from activation of regulated necroptosis, which could include contributions from both necroptosis and mitochondrial permeability transition. Treatment with the drug cyclosporine, which represses the mitochondrial permeability transition effectors cyclophilin D, improves tissue survival primarily by inhibiting necrotic cell death rather than its additional function as an immunosuppressant. Now we'll see necroptosis based therapy. Recently, necroptosis based cancer therapy using a distinctive molecular pathway for regulation of necroptosis has been suggested as an alternative method to overcome apoptosis resistance. For instance, necroptotic cells release highly immunogenic DAMPs, initiating adaptive immunity. These dying cells can also activate NF kappa B to express cytokines, recruiting macrophages. As of 2018, little is known about negative regulators of necroptosis, but CHIP CFLIP, and FAD appear to be potential targets for necroptosis based therapy.